All right, Living Environment fans, I'm here with my buddies Francis Crick, James Watson. You may remember them from the discovery of the structure of the DNA molecule. And today is the last Bellringer of the second quarter. So let's make it worthwhile. All right. Hey now.
Mr. Kovacs class. It's Mr. Kovacs class.
You might learn a ton.
It's Mr. Kovacs class.
It's interesting too.
It's Mr. Kovacs class.
And he's super cool.
It's Mr. Kovacs class.
Yeah, hey, and of course Watson and Crick were responsible for figuring out that the double helix structure of the DNA molecule, right? It's got sugar and phosphates on the sides of the ladder, but the rungs have what we call the nitrogen bases, or the nucleotide bases, and there are 4 of them, and they always match up: A with T, G with C. You know, it's interesting. Just gave the Living Environment Regents in January the other day, and there's a question I'm going to ask you right now and see if you can answer it. It's a Regents style question. Let's say you looked at a sample of DNA and it was 18% adenine. What percent guanine would you have? Yeah, that's right, because it's going to be, if it's 18% adenine, it's 18% thymine, right? So that's 36 total percent. That means there's 64% left over, split evenly between the guanine, right? They have to match. So the answer was 32%, right? Guanine. So that's, yeah, that was a Regents question. Let's see, what else? We're in lab this week. We're pulling DNA out of fruit in the battle of the fruit. Let's see what that's all about. Of course, you've got to take the mass, right? We're taking the mass of the fruit before we do anything to it. And then we're going to pull the DNA out of the fruit, and then we're going to calculate the ratio of the mass of DNA to the mass of the whole thing. That should give us an idea of how much DNA is in each cell, more or less. No, it's not Baja Blast Mountain Dew. It's or blueberry flavored Kool-Aid. This is your DNA extraction buffer, which was a mixture of soap, salt, and water. And the job of this mixture, right, is to basically break apart cell membranes, and any membrane, so that the insides of the cell and the organelles inside the cell will fall apart into pieces, and then we can get those pieces into a solution and pull the DNA out. Smushed up your DNA in a baggie, added that solution, and then you ran it through a filter, right? Filter, and collected it in one of these New York State issue kind of cups. Right? And then we use rubbing alcohol, isopropyl, cold rubbing alcohol, to pull the DNA out of solution, and we get some DNA. Then we weighed that. And then we got this nice, let's focus on that, yeah. This nice goopy bit of DNA, and we put it on the balance and weighed it. And then we did the ratio. So let's see if the strawberry was 30 grams, right? And then your weight of your DNA was 0.30 grams. The ratio is 0.30 divided by 30 grams. Let's see, that's going to be 0.1 over 10, or 1/100th. So we could say that that DNA was 1/100th the weight, or 1% of the weight of the strawberry is DNA. And next you compare it to a banana. You do the same routine, right? Controlled variables. The independent variable in this case is the type of fruit. The dependent variable is the ratio of DNA, right? The weight of the DNA compared to the weight of the fruit. You know, that technique that we use in class is similar to what they use in those, those crime shows. When they find DNA at a crime scene, they need to collect specimens that have, or tissue that has DNA in it, and then they have to use the DNA extraction buffer and isopropyl rubbing alcohol, right, to pull the DNA out of solution. And then what they do is they cut the DNA up into fragments using what's called a restriction enzyme. We'll get into that later. And they put that solution in a gel and then run electricity through the gel. And what happens is the smaller the fragment of DNA, the farther it moves. So you get this, what we call our DNA fingerprint. And everybody's DNA is unique unless you're identical twins. Right? And so this is how we use DNA technology. Is the same that you use to get a straw from a strawberry, right? Used to solve crimes, like finding out the DNA fingerprint.